about Professor N.S. Deodhar first, because some of us have interacted with him, but many of our students did not get that opportunity. So I'll be reading about Professor Deodhar, and then he will read the citation. The Professor N.S. Deodhar Award was instituted on 5th March 2020 by the department in his honor as one of the founder members of the then Biometry, Health and Nutrition Department of Pune University. An able team that included Professor Deodhar, Professor Mutatkar, Professor Bhushan Patwardhan and Professor Anita Kar, under the guidance of Professor Shekhar, nurtured and built this school to its present stature. As referred to as the architect and father of public health in India, Dr. Deodhar has been the guiding force behind the departments since its inception. As a public health professional of eminence, he spearheaded the cause of public health for over 6 decades. His contribution to prevention of disease and health promotion approach was instrumental in developing the discipline of preventive and social medicine in India. Comprehensiveness, critical thinking, and interpretation and down-to-earth practical approach were characteristics of Dr. Deodhar as a person and as a public health researcher. His contribution is not limited to the public health, epidemiology, primary health care, but extend beyond to health administration and management. His work received international recognition by his election as a member of the International Epidemiological Association to represent the Southeast Asian region, and later award of honorary membership of IEA in year 2000. He had held several prestigious positions with academic rigor and administrative flair. Professor Deodhar headed the Department of Preventive and Social Medicine at BZ Medical College and upscaled it to an advanced unit. He was part of more than 185 committees and commissions at national and international level during his illustrious career. He was invited by the World Health Organization to develop the first national program for training medical students in community medicine, leading to MD degree in University of Colombo, Sri Lanka. He was the chairman of 3rd International Congress of World Federation of Public Health Association, where representatives from 51 countries participated. Dr. Deodhar has contributed immensely to the policy making as a member of the National Commission on Population and the Planning Commission of India. His proficiency in public health is exemplified in his incisive studies on plague outbreak in 1994, contribution in eradication of smallpox from Maharashtra, studies on his development of national programs such as HIV/AIDS, tuberculosis, malaria control and polio eradication program. His contribution, published in more than 260 papers, are of great value to scientists, administrators and policymakers. Some of the renowned national and international honors bestowed on him for his outstanding contribution include PHFI Lifetime Achievement Award, Ved Prakash Memorial Oration in 2008, Best Citizen of India Award in 2011, Jeevan Gaurav Sanman Patra in 2011, Dr. R.C. Tiwari Memorial Lifetime Achievement Award in 2015. We are blessed to have had his guidance in this very school. Today, we feel happy to honor Dr. Shekhar Mande with Professor N.S. Deodhar Oration Award for the year 2024. I request you, sir, to please come up on the stage. Citation. So please answer. The citation that will be awarded to Professor Shekhar Mande, I am going to read it. Department of Health Sciences, Savitribai Phule Pune University feels honored to award the Professor N.S. Deodhar Memorial Oration to Professor Shekhar Mande, former Director General, Council for Scientific and Industrial Research, former Secretary, CSIR, Government of India, and distinguished professor of Savitribai Phule Pune University. Professor Mande is being awarded this citation for his outstanding scholarship, scientific contribution, and strategic leadership in advancing Indian science. As a structural biologist, Dr. Mande's pioneering work in computational system biology has significantly advanced our understanding of the structure-function properties of bacterial proteins, particularly those derived from Mycobacterium tuberculosis. His research has been instrumental in the field, and he was rightfully awarded the Shanti Swarup Bhatnagar Prize for his outstanding scientific contributions. Professor Shekhar Mande's leadership at the National Center for Cell Science, Pune, was transformative, leading to significant advancements during his tenure as director. His role was particularly crucial during the COVID-19 pandemic, where he chaired the COVID-19 strategic group of CSIR and spearheaded efforts in disease surveillance, development of novel diagnostics, research on therapeutic interventions, development of hospital-assisted devices, and managing supply chain networks. Professor Mande, inclusive approach and leadership were instrumental in developing CSIR laboratories. His efforts to foster transdisciplinary research, collaborations by engaging industry, policymakers, and international institutions are commendable. We feel honored to present Professor Deodhar Memorial Oration Award of 2024 to Dr. Mande. Over to you, sir. Thank you.
Thank you, Ravi. Professor Parag Kalkar, Professor Prof, University of Pune. Professor Abhay Gudale, Health Department, Health Sciences. My good old friends, Bhushan, Anita, parents of Suparna Bakshi, Mr. and Mrs. Bakshi, Professor Bradford, Dr. Gupte, all the friends and colleagues present in the audience here. Firstly, I feel very humbled to be present on the occasion of starting Dr. Suparna Bakshi Fellowship by her parents here and recalling her association with the department. And I would like to thank Mr. and Mrs. Bakshi for your gesture, and I echo the sentiments expressed by Professor Kalkar that this would be a long-lasting memory of her in the university. Secondly, I also feel extremely privileged to be able to give Professor Deodhar Memorial Lecture to all of you. Both the previous speakers of this lecture series are present in the audience, and I feel extraordinarily privileged that you have chosen me for this honor today. So thank you very much, the committee and the faculty members of the department for bringing me here to give this particular oration. The other day when I was talking to Girish about what possibly I could speak, and Girish suggested that because during the COVID period, I happened by accident to be in Delhi, and what the CSIR did at that time, I could recall some of the aspects of how government of India, and I was a small part of that particular effort, how we actually contributed in the COVID-19 mitigation strategies at the government level. And some part of that, I will present the view of CSIR to all of you in this particular respect. How the science and technology community, which was essentially spearheaded by CSIR, fought the COVID-19 pandemic. So to go to that, we first recall what COVID-19 was about, what we know today about COVID-19, and then we will go back to our memories starting from 2020. Today, we know that COVID-19 was caused by this dreadful virus, what we call as SARS-CoV-2, Severe Acute Respiratory Syndrome coronavirus, the second kind. The first epidemic was there about 20 years ago, but it did not become as serious as the SARS-CoV-2. In December 2019, clusters of pneumonia-like cases were reported from the Wuhan province of China. About mid-December to end December, And by end December, I think people had been alarmed about the rise in cases at the Wuhan province. And by early January, the first week of January, it was discovered that this disease is caused by a virus, and the virus was identified to be of SARS-CoV type, the coronavirus type, and then it was named SARS-CoV-2. Then the first genome sequence of this became available on 7th of January 2020. The first of the cases in Europe and US, they were reported in mid-January. So you can see within 15 days from China, the virus had already traveled to places like Italy, where there was a very serious outbreak of an epidemic. And in the US, reported the first of the cases on 17 January 2020. In India, we reported our first case on 31st January 2020. A nurse who had come from the Middle East to Kerala, she was the first one to carry the disease into India, the first reported case into India. And that's where all our alarms were raised. It so happened that on 7th February 2020, I happened to be in Pune and I gave a talk in the Gandharva auditorium in the center of Pune. And in my talk on 7th February, although we were only 7 days old in our first case, I did mention in my talk, and it's available on YouTube, saying that we are living in the coronavirus scare on today's day. So the enormity of the disease was apparent even at the beginning of the pandemic. And that's how all of us were prepared for that. So if you look at the timeline, as I said, early 2020, January 2020 was the critical period and the first of the cases were being discovered. As I said, in India, 30th January was the first case reported. It so happened that CSIR, which is a conglomeration of 37 labs, a chain of 37 labs across India, which work on many different fields, not necessarily only diseases, but also on aerospace, mining, oceanography, and so on and so forth. The directors of the 37 labs met in Goa on 25th-26th February. And it is at this directors conference our first discussion on the mitigation strategies for COVID took place. Remember, the world had not yet recognized that what we are looking at is a pandemic. The world was still waking up to the reality that we are on something which is very important, something which is probably very devastating. But we had a detailed discussion on 25th-26th February in the directors conference, and as luck would have it, 15 days later, WHO declared on 11th March COVID-19 as a global pandemic. Now you have to remember, the public health experts here would know the difference between an epidemic and a pandemic. And this was one of the first pandemics that we have been encountering in a long period. Although it had been accepted, it had been expected that the next pandemic is just around the corner. But nonetheless, when it hit us, we were least prepared. The entire world, including the most advanced countries, were least prepared for the pandemic that we were going to face. We did not know what kind of pandemic we were going to face, and that came in the form of COVID-19. Government of India, and all of us were involved in the discussions leading to that, announced the first lockdown on 24th March. Now the lockdown has been discussed in press, media, everywhere. The lockdown has been criticized by the critics of the government, but there was a very well thought strategy before announcing the lockdown. And the strategy that all of us had collectively discussed in the Ministry of Health and at the Prime Minister's office was essentially to prepare ourselves with everything that what we wanted to do to tackle the pandemic. Right? And CSIR at that stage, CSIR as I said, it's a collection of 37 labs. We formed a COVID strategic group, and our first meeting was held at 5 p.m. on 29th March, online meeting. And from this day, next 6 months, the meetings of the CSG group were held every evening, online every evening. They were core members of the group, and they were members of other groups whom we actually used to call from ICMR, from the principal scientific advisor to the government, from Niti Aayog, from other organizations who were involved in fighting off the pandemic to our meetings. These meetings would typically start at around 6 o'clock in the evening and would go on till 9, 10, so on and so forth, depending upon what kind of things were discussed. And based on these meetings, what we actually did an assessment, the first assessment was that, what did we know about SARS-CoV-2? We knew that a country had very limited diagnostic capability and capacity. We as a country did not have, I will come to you, I will tell you how the diagnostic capability was enhanced in literally the lockdown period. There was no point of care diagnostics. All that was known that RT-PCR was the only way that we could diagnose the virus. There were no vaccines, there were no therapeutics. We knew that the virus was highly infectious. We knew that it causes respiratory failure in critical patients. What we knew very early in the pandemic was that the virus causes lower respiratory infection, and in healthy humans, the virus is cleared in about 7 to 10 days from the body. The virus is cleared, right? I mean, there is an initial phase of rapid multiplication of the virus in our body, but very fast the body responds, and the virus is cleared by the 10th day. Your viral count is almost zero. But then a strange thing happens on the 10th day. And at that time, the body hyper-reacts. The body's immune system, what we call the cytokine storm, that takes place. And that goes on damaging the lungs. And that is the one which is fatal in most of the cases. So almost everyone who died of COVID, of SARS-CoV-2, died because of the hyperimmune response and not because the virus killed any of the cells in the body. That's one fact that we have to keep in mind. But this was not known till very late. And of course, the biology of that was known in early March 2020. In CSIR, when we assessed the situation, we knew that we have very wide experience in things like genome sequencing and bioinformatics. We had a great experience in diagnostics. We were in the fountainhead of generic pharma industry in the country. Generic pharma industry started in CSIR, and we had been supporting that over the years through the country. We have expertise in design and engineering of medical devices, and we had a capacity for tackling infectious diseases. So that's where our strengths and that were the unknowns that what we had. So taking stock of the unknowns and taking stock of our strengths, we decided a strategy to tackle the disease very early on in one of the early CSG meetings. And we decided we were going to work on 5 different verticals. And the 5 verticals that we decided to work on, and as I said, they are not CSIR alone. We also included IITs, IIC, ICMR, Department of Biotechnology. Many of them joined in our efforts in doing this. And the 5 verticals were surveillance, diagnostics, drugs and vaccines, devices, and supply chain logistics. And this I would like to come to this one by one in the next few minutes. And in the process, over the next 1.5 years, we generated about 100 odd technologies, and the technologies were transferred to some of the best known companies in the country, and these companies were able to scale up the technologies and they implemented this in the field. What we see today, in no time, in a few months, most of the technologies were visible in the field. And all the companies had come forward, whether it be Reliances or the Tatas or the Sun Pharmas or whosoever, they had implemented in the field and they were being used all over the country. And that's how the country actually stood up as one single entity to fight the disease. And therefore, you would actually see, in retrospect, the impact of COVID on India was serious, but not as serious as that, for example, in Western Europe or US or in advanced economies. Now, this fact actually is not digested around the world. This fact is not accepted by some of the advanced economies. But if you look at the total number of people living in India and how many people died, that ratio of India is much lower than any other advanced economies around the world. So India's response is something what all of us ought to be very, very proud, the way we tackled COVID-19. So I will come to the 5 verticals one by one and tell you a little bit about what these were. The first one was on surveillance. The first thing that we actually put in place, as I said, RT-PCR was, many people did not know, even in medical hospitals in the country, people did not know how to do RT-PCRs. So our first strategy was to train large number of professionals who would do RT-PCRs. So our labs, for example, in Hyderabad CCMB, they gathered the entire Telangana and Andhra Pradesh fraternity together and started every week training a batch of people who could competently do RT-PCRs. RT-PCR machines did not exist in many places in the country. And most of our biology labs, for example, if you look at National Center for Cell Science here, would have something like 10 RT-PCR machines. In ISCB Calcutta, we had RT-PCR machines, and we donated RT-PCR machines to the hospitals. The Army Hospital in Calcutta did not have a single RT-PCR machine. That was donated by our lab, Indians of Chemical Biology there, and we went and taught people how to do RT-PCR. And this way we could actually very rapidly generate a capability in a large number of professionals who could do RT-PCRs. In remote areas, for example, Ladakh, obviously the healthcare system is not as developed as in the rest of the country. And we airlifted during the lockdown, as I'm showing you here, then Air Force plane. It is carrying all the machines for isolating RNA and doing RT-PCR in 2 locations in Ladakh, in Kargil and in Leh. So these are actually being audited. So large number of people were trained in CSIR laboratories, whether it be CCMB Hyderabad or IGIB in Delhi or ISCB Calcutta, in different corners of the country, large number of people were trained in doing RT-PCR and scaling that up very quickly. We came up also with the standard operating principles for each of those. That's the first thing. The second thing, we wanted to see how fast the disease is spreading. And one way of testing that was put in place serosurveillance. Somebody mentioned serosurveillance here. It actually was begun with a centralized initiative in Delhi. We actually started serosurveillance. ICMR did their own serosurveillance. We did several serosurveillance. And we did multiple serosurveillances every 6 months or something like that. And that told us how the virus is actually spreading across the country. So these are actually some of the papers that came up. There's also a paper in the journal Science about India's serosurveillance and how the virus was actually spreading very rapidly. But apart from that, surveillance at different levels also was necessary. And one of those was, of course, the genomic surveillance. Now this is one place where we face challenge, because some of the other agencies which are the upholders of India's medical system were not very keen on actually putting genomic surveillance in place. But we put down our foot. In fact, a meeting that was chaired by the Honorable Health Minister then, myself, and the Director of Institute of Genomics and Integrative Biology, we put our foot down and said that we are going to do genome sequencing at a very large scale. And we put actually in place practices in states like Kerala and in Maharashtra that every week from every district we will receive samples and we will actually sequence them. And that's how variants later like Delta and all were discovered in the country. Nonetheless, the CSIR labs actually stood up and eventually we formed a consortium, today what we call as INSA-COG, a consortium of multiple labs in which we went on sequencing multiple variants of the virus, and that actually gave us confidence how the disease was spreading and if the mutants were arising in different places, what kind of strategies could be adopted there to do that. I will come to CVS surveillance very soon, but we also were the first people in the world, remember not WHO, India was the first country in the world to discover that virus is airborne. WHO all the time had been saying that infected people when they cough, they emit what are called as kind of aerosols, the particles that float around with size of roughly about 5 to 15 microns. These particles very quickly settle on surfaces, such as wooden surface or soils and all. And other people who touch that contaminated surface eventually will get the infection by touching that particular one. That of course is true. But what is more important is that virus remains intact in aerosols and aerosols remain suspended in air for a long time. And that is what an airborne infection essentially is. And we are able to do these experiments in our labs, in labs such as the Central Building Research Institute, believe it or not, a civil engineering lab, where there was an aerosol facility and where there was airflow facility, we were able to show that indeed the virus actually remains suspended in air. And then we went to the samples to the Institute of Microbial Technology in Chandigarh, which had a GSA3 facility, and showed that indeed the virus entrapped in these aerosols was live virus and could spread for a long time. WHO team took another 8 months to recognize the virus was airborne before we had actually started talking globally about this. And severe surveillance, as I said, we are very proud that we put in place the severe surveillance very quickly. And in the initial period, of course, a large number of studies that came up. For example, in early 2020, in mid-2020, we were able to show that in Hyderabad, about 6 lakhs people were infected between July and August 2020. That's very early during the pandemic, the first 6 months. Taking the samples from sewages, hospital, residential area, and so on and so forth. And one of those severe surveillance samples, that study had just been published about a week ago from our group in National Chemical Laboratory here, in collaboration with Bishop Medical College and so on and so forth. They have published a very nice study only about a week ago that how severe surveillance actually tells you what variants of virus are actually emerging either in environment or in immunocompromised patients. As far as the aerosols are concerned, if we know that virus spreads through air, through aerosols, we must have a strategy to control that. And based on that, how long the aerosols would remain suspended in any air, we came up with guidelines saying that if the rooms are enclosed and if you close all the windows and doors, the probability that these particles would remain suspended in air is much higher. If you open all the windows and doors and if there is cross ventilation, the probability that these particles would remain suspended in air is lower because of ventilation. In open areas, the probability that the particles would remain suspended in air and actually would go and infect other people is much lower. In the air-conditioned areas, especially during winters or in summers, if the air that is coming into the room, if that is not sanitized and if that air is circulated, the probability of infecting others is higher. And that typically happens in places like air-conditioned rooms, in airlines, aeroplanes, it happens in trains and so on and so forth. And to do that, we came up with a strategy how to sanitize air that comes into the room, right, in the air-conditioned rooms. And the first of the strategies that we did was a UVC essentially. And we went and installed this in the Parliament of India. So the Lok Sabha and the Rajya Sabha, both of them, we calculated how much airflow was going in, and based on the computational fluid dynamics analysis what we had, we went and sanitized this. This was all validated based on the airflow measurements that we had done in the Institute of Microbial Technology in their BSL facility. How much UV exposure is needed to inactivate the virus before the air comes into the room. And today, this, if you happen to ride any of the Mande Bharat Express in the country, you would see at the entrance, it would actually be a warning that Mande Bharat Express has a sanitization strategy which is used something like this. And that is not true for SARS-CoV-2 alone, it is true for any other airborne infection. And therefore, our Mande Bharat Expresses are the first of its class in the world where the air that is circulated inside is actually sanitized for any of the microbial infections. I think we should be very proud. And all these results have been published in scientific literature. We came up, of course, with the guidelines that were circulated widely by the government on what should be the ventilation in the residential buildings in that particular manner. Coming to diagnostics, as I said, RT-PCR was the only diagnostics that was available. And very quickly, many people realized that many other kind of diagnostics could be done. For example, the diagnostic kits that are distributed at home, they essentially test whether you have an antigen circulating in your serum. So they're essentially antigen based. But we also came up with a diagnostic strategy based on what is called as CRISPR-Cas. And India became globally only the third country which had evolved this CRISPR-Cas technology based diagnosis. We called it FEBUDA, which is an acronym for FN-Cas9 editor lane uniform detection assay. And there is a specific thinking why we called it Feluda. Because the Harvard group, which had done a CRISPR-Cas based diagnosis very similarly, the Harvard group had named it Sherlock, and the University of California Berkeley group, the people who eventually got Nobel Prize for working on CRISPR-Cas, that group had named it Detector. And therefore, we had actually to come up with a name, and what better name than Feluda. And those of you know what Feluda is, of course, you would have read Feluda books, you would have seen Feluda movies, and actually that. And in fact, hilarious incidents occurred when I made a presentation to Honorable Prime Minister, and Honorable Prime Minister did not hear me correctly. He thought I was calling it Faluda, and he said, why do you call it Faluda? So I gave no solution of Faluda, it's Feluda. Then he understood what it is. Alright. But we also came up with something which is very ingenious, is that a virus is transported from the collection site to the detection RT-PCR site in a viral transfer medium. And that leads to multiple problems. If you take actually a swab, dip it in viral transfer medium, seal it, and then send it to place where it is to be RT-PCR, you have to first isolate the RNA and then do RT-PCR. We said all of it is not necessary. And there is of course the danger that if the tube breaks, then the viral transfer medium can spill. We came up with what is called as the dry swab direct RT-PCR diagnostics, in which you did not need viral transfer medium and you did not need to isolate the RNA. Right? So with this technique, we were able to do the test much more faster, and we were able to do it at about half the cost of standard RT-PCR, of isolating RNA and so on and so forth. And all the expenses on isolating RNA from automatic RNA isolation machines were present. And we quickly tied up, as I said, for every technology that we are developing, the Feluda was manufactured and scaled up by Tatas. You would have seen multiple places in the country. For example, Bangalore airport had a counter set up by the Tatas, saying that any person arriving in Bangalore airport, they were actually doing Feluda tests. Feluda was actually circulated across the entire northeast, across Himachal, across Uttarakhand, because remote places had no means of doing RT-PCR. They had to wait for 7 days at times for collection of sample, sending to a place of doing RT-PCR and sending the results back. Our buses actually went to all the places with Feluda, and we were able to diagnose in many of the remote areas at a particular point of time. Similarly, the dry swab RT-PCR was available in all the Apollo hospitals across the country, and the Spice Health. The Spice Health essentially is a subsidiary of the Spice Jet, right? And as I said, the genome sequencing also was set up by Spice Health at Delhi airport and multiple places in the country where people could give their sample and have the SARS-CoV-2 sequence there. As far as the drugs and vaccines were concerned, we conducted large number of trials, and one of the first drugs that was actually came to the market, it is an anti-flu drug called favipiravir. And we were able to actually quickly do limited clinical trials because they repurposed drug, and we did clinical trials for this, and Cipla manufactured and marketed in the country. So it's available at a very large scale in many places, many hospitals gave this one. And of course, as I said, I must say here that remdesivir was announced around the world as a very effective inhibitor. I was given the responsibility personally to bring remdesivir to India, and I negotiated with company in Gilead, their CEO, and how do we bring the drug to India without violating their patent rights and so on and so forth. And in Gilead, with our persuasion, we were able to give the license to multiple Indian pharmaceutical companies, and we were actually able to make remdesivir in India at a much larger scale. But apart from that, we undertook large number of clinical trials with multiple the pharmaceutical companies, whether it is Sun Pharma or Cadila and so on and so forth, and we actually started repurpose clinical trials. We also did trials for some of the Ayush populations. For example, Ayush 64, Ashwagandha, Guruchi and Pipli are the ones that we did along with Ministry of Ayush, and this clinical trial, some of them were conducted in India. As far as the vaccines are concerned, we actually stated that there is already a vaccine effort that was very strongly going on by some of the private companies like Bharat Biotech and Serum Institute. So we did not want to get into the vaccine area apart from that. But whenever they had difficulties, they approached us. For example, Bharat Biotech had tremendous difficulties in making an adjuvant. So the adjuvant that they were using was something called a TLR79 agonist, a toll-like receptor 7 or 9, the agonist of that is used as an adjuvant in the Covaxin vaccine. And they had difficulties in making that. So they approached us, and our laboratory in Hyderabad, the Indian Institute of Chemical Technology, made that adjuvant in kilograms scales. So every week they used to make a few kilograms of the adjuvant and used to give it to Bharat by day. And any one of you here has taken Covaxin. You have the adjuvant taken from the CSIR lab, which actually made that in a clean facility in the CSIR lab. You know, something like that we actually doing. But we also wanted to get into devices because there was a need for actually doing that. And as all of you know, that the pandemic, when it hit us in March, the country did not have capability to make PPEs. The country used to import all these swabs. The country could not make VTMs on its own, viral transfer medium on its own. All of it was imported. Can you imagine the nasal swabs were imported from China? Can you imagine that we did not have capacity to make PPEs? And we have to very quickly make sure that our companies start making this. And we put in place a strategy where on a large scale, all this can be made within India. And within 6 months, the country had become completely self-reliant on making all of this in India. Whether it be PPEs, whether it be swabs, whether it be mediums, whether it be test tubes or whatever, all of it was now being made in India. And here just to show an example from NCL here, right here in NCL Pune, who actually made these swabs. They made from one of the polymer materials, and it was then transferred to this and has been actually used in several labs. This was actually made and distributed all across India. We took a conscious decision because one of the persons of our core strategy group was Dr. Anurag Agarwal, who is actually himself is an expert of pulmonary medicine. And he told us that we took a conscious decision that we don't want to make an invasive incubator in which the tubes and all are inserted. What we want to do is that we want to make an incubator which would avoid taking patients from a normal ward to ICU. Typically, invasive incubators are essentially used in the ICU if the patient is really in a very critical need of oxygen. But to avoid patients taking to ICU itself, if we are able to improve the lung health before that, that actually could be done using ventilators such as what we call as the bi-level positive airways pressure or BiPAP, this kind of ventilators. And very quickly, because we had labs like National Aerospace Laboratory who work on civilian aircrafts and have for years worked on airflow management systems, they were actually able to make a ventilator. Can you believe in aerospace laboratory? Because of their expertise in airflow management, they were actually able to make this. And a record of 36 days, they made this particular ventilator. We went to clinical trials. We made clinical trials in the Manipal Hospital in Bangalore, and we approached the DCGI at that time, and we got emergency approval for using this. And in several thousand copies of this were made, and they were distributed all across India to multiple hospitals. Remember, these are given to patients not in ICU, but before going to ICU, to reduce the burden of going patients into ICUs. And that was a strategy that we actually adopted very consciously at that particular point of time. When the country actually started feeling the heat on oxygen, it was roughly the time about March 2021. The Delta wave was just starting. And at that time we realized that we are going to fall short of oxygen. And at that time, Indian Institute of Petroleum in Deodhar, they came up with, they already knew how to enrich oxygen from air, and using zeolites and all, they could actually separate nitrogen and oxygen, and they could get actually medical grade oxygen. And first of the plants was successfully commissioned along with NCL Pune. So Indian Institute of Petroleum Deodhar and NCL, the first of the plant was successfully commissioned in May 2021 at the Avni Chase Hospital. And at that time, DRDO and us together, we came together and we said that we are going to make a large number of such plants such that every district in India would at least have one medical grade oxygen plant, every district. And we made thousands of these plants and distributed very quickly, and oxygen was actually then made across every district. And in cities, of course, multiple hospitals now have actually bought these oxygen management plants and they have this plan. But apart from that, oxygen for a personalized level, so this oxygen goes into the oxygen lines in hospitals. This is a very large plant. But even personalized oxygen plants actually could be be made, and NCL actually made oxygen management units such as this, which should be personalized for a person who is admitted in the hospital. It is about the same time in 2021, during the Delta wave, we realized that we are running out of hospital space. All of you remember that hospital beds came at a premium. Right? All hospitals in the country were full. It was a pathetic situation. The Delta wave was devastating on India. And at that time we realized that if we don't generate enough hospital space in the country, we are leading to even more catastrophe. And we had two labs, the Central Building Research Institute in Roorkee and the Structural Engineering Research Center in Chennai. They together came up and said a prefab building can be made at a notice of 7 days. If you have prefab material available, we can make a fully functional hospital at a notice of about 7 days. And the material can be carried on your shoulders. You go and assemble the entire building at one particular place. And believe it or not, we made this kind of hospitals. For example, DRDO made hospital at near Delhi airport, about 1,000 bed. We made a hospital in Safdarjung Hospital in their car parking lot. Safdarjung Hospital had run out of space, so the car parking lot was used to make hospitals like this. But many remote places, Himachal for example, Himachal had very shortage of hospital space. So we went to multiple places in Himachal and made those things. What I show you here is a makeshift hospital. We call it makeshift because it's made from prefab material, but it's a fully functional hospital with all the oxygen lines, with a functional ICU and so on and so forth. And this is the one that is there in Kangra, for example. If you happen to go to Kangra, there is a 100-bed hospital that we actually made there. We made a 500-bed hospital in Bhatinda, and so on and so forth. Multiple companies came forward that can we make it in our area. For example, Garware, they approached us and said that can we make it in Nandurbar, in Maharashtra. We said, of course we can do it. And in Nandurbar, we went there and built 500-bed hospital in Nandurbar, along with Garware's corporate social responsibility funds, and so on and so forth. So, large number of places in the country, we are able to generate hospital space. Although these are called makeshift, these are good at least for next 25 years. So, we have fully functional hospitals across India now in very remote places such as this. So, as you can see, the entire approach has been very holistic. Whatever we have been needing, unfortunately, at many times we are not able to foresee the need of some of this, but whenever we saw the need, we are able to meet that. And to be able to do that, one has to make sure that one's supply chain is a robust supply chain. Right? For example, if you want to make diagnostics, every chemical that goes into diagnostics, RT-PCR we will do, and RNA isolation kit. If you want to do a PCR, the nucleotide sequence that you name, the machine that you name, the buffer that you name, all of it has to be available. And has to be available at the drop of the point. Or you should know where that material is available. And this entire thing actually burns down to what is called as supply chain. If you want to make a hospital, all your material to make the hospital, gas lines, the wall materials, the floors, everything has to be made available. And the entire supply chain has to be made available. And supply chain, of course, if it breaks at any point of time, that once again is a disaster situation. And that supply chain, what we did, we had a very successful app that we had made earlier, before COVID. It is called Kisan Sabha. And this Kisan Sabha supply chain essentially works with the farmers. That farmer produces something on the farm, and the transporters who come there pick up this produce on that particular farm, and the transporters take it to a market and sell this particular produce of the farmers in the market. So this is supply chain, right? And this app actually connected all the 3 dots together: the farmers, and the transporters, and the markets. And this particular app, therefore, we were able to repurpose very quickly for making a supply chain for that, what is called as the Arogya. So Government of India immediately adopted this, Ministry of Health, and we actually had a supply chain app on a mobile that any point of time you want a PPE, you find out what is the closest place that you can get a PPE. Anytime you want the swabs, then you can get a swab. And all the diagnostic centers, all the medical chemical shops and all were actually onboarded on this particular app so that people could very quickly connect with each other and get it. So essentially, what actually meant is that the response to COVID in India was extremely holistic. I have given you examples only from CSIR because of my perspective from CSIR. But similar approach, as I said, whether it be ICMR or Department of Health, Ministry of Health or Department of Biotechnology, and all of us were talking to each other practically every day. So almost every day or every alternate day, somebody like Dr. Balram Bhargav, Dr. Renu Suru and I, we used to talk to each other. Every alternate day we would go to Niti Aayog and talk to Dr. Vinod Paul. So all of us were in constant touch with each other on the emerging issues in COVID-19 in India. And actually we are assessing what do we need on that particular day and how we are actually going to make a robust system that those things are needed at a very short notice. So essentially, what we learned from the entire pandemic are multiple things. Number one is that we need to connect with almost every stakeholder in society. And the stakeholders included the industry. I had written an email, personal email, to chairman of all the major companies in India, whether it be Tata, whether it be Reliance, or something so on and so forth. And I was told that my mail to Mr. Mukesh Ambani reached in the evening at around 9 o'clock, and he called for a meeting of all the Reliance subsidiaries and the chairman and CEOs and all at 10 o'clock in the evening, and placed my mail in front of them and said that we have to work with the government. Mr. Shekhar, the chairman of Tata Sons, next day he came in an online meeting for 1 hour with us. And at that time, we were actually worried that the Delta was reaching Mande. And we were extremely worried. There's a very grim situation that around end of March, Delta wave had already begun, March 2021, and we were worried that Delta reaches Dharavi. And we would be washed out. As a country, we will not have a face to show anyone if there is a large fatality in Dharavi. And we requested the industry people that please go and make sure that people stay away from each other. They don't crowd, they don't make crowds. If they have to wash hands, please make sure that foot-operated washing machine wash basins are made available. If they want to go to common toilets, make sure mark the places in which they maintain at least 5 feet from each other. And all this, actually, the healthcare workers on the ground were doing that. So what was needed was a close interaction of the government, of all the R&D organizations in the country, all the industries and the societies. So a connect with all the stakeholders was necessary to handle the pandemic at a particular stage. There is a need for the scientists and technologists to work closely with the social sector, and this has been one of the biggest learnings from the pandemic. And what happened during the lockdown, there were many people who actually started migrating to their villages, and we have to make sure that these villagers don't carry, number one, COVID with them to their villages. Number two, because the situation, the social situation was so stressed that they should also be provided with food, water and shelter for the night and so on and so forth. And it's at that point, the entire country rose up. And it's so heartening to see that how the entire country as a community, as society rose up, and people who are going back to their native places, whether it be Bihar, whether it be Northeast and so on and so forth, all the way from, let us say, Mumbai or Pune, they did not face the same crunch had the society not risen up on that particular occasion. It was an extraordinary situation. And it's a human learning that all of us must realize that how we as a society rose up to that particular challenge of people who are going back. Third lesson, we want to reduce imports and be self-sufficient. And this actually I have highlighted in my supply chain logistics issue, that if the supply chain is broken at any point of time, to give you an example, when we were doing oxygen enrichment, the zeolite that is needed to enrich oxygen from air is available only in France or a couple of companies in the US. And if there is delay in getting that zeolite here, we will not be able to make oxygen very quickly, the medical grade oxygen very quickly. And for that purpose, we decided that zeolite must be made in India. And NCL very quickly got into the work, and NCL actually made that zeolite and transferred the technology to a company who could make the particular zeolite at a large scale. So every small thing that goes on making anything, whether it be device whether it be a PPE or whether it be any kind of other equipment that is needed, that must be available in India. If you have to do RT-PCR, the oligonucleotides or the short nucleotides have to be made available in India. We cannot depend upon importing from anywhere else. And that's one of the learnings from the pandemic, that how do we reduce imports and be self-sufficient from all purposes that we have. And then we must map technologies for local use. As I say, as very popularly said, that science is global, the scientific discoveries are global, but the adaptation to technologies are always local. Depending upon the situation that is there, the technology has to be adapted. And lesson number 5 for future is Atmanirbharta that we are talking about is not for today, it is for the future. If the country wants to actually become a major global power in the next 25 years, the country must learn to become Atmanirbhar 20 years from now, not today alone. So all the technology that are happening in the world today, or the development that are happening today, India must be in step or in sync in developing those technologies. And lastly, of course, this is not the last of the pandemics that we are seeing. There are many more pandemics which are around the corner. In which form they will come ahead, we don't know. Right? The nature of the pandemic on today's date cannot be predicted. It could be communicable, it could even be non-communicable. What pandemic we are going to face is uncertain. But we should have a system in place which can deal with any such contingent situation that arises tomorrow. And that's the major message that I would like to give to all of you, that we must be prepared for any kind of contingent situation that arises tomorrow. I would like to thank in the end whole lot of government machinery, different ministries of the government. I would like to thank all the industries who work with us, just representing some of them who actually worked at a particular point of time with us. I would like to thank all the society in general, the Indian people, really those, whenever we made an appeal to them, not to panic, not to do this, not to do that, wear masks. The mask wearance compliance in India was way more than most of the other countries. The vaccine compliance in India was way more than many of the developed countries. Right? And that's because all of us were continuously talking to people and saying that please do this, please do this, please do this. And that really helped. And it came from the highest level in the country, that urging people to wear masks, urging people to get vaccinated and so on and so forth. And we should be very proud that we were actually able to tackle the pandemic in that particular sense because of the entire collective effort of what I have said. I would not say very effectively because there are still a large number of casualties, but I would say that the impact of pandemic would have been much worse if we had not done all of this. Okay? And to that extent, we should at least pat our backs if we have done reasonably. Thank you.
Thank you, sir, for a wonderful presentation on CSIR's contributions during COVID-19 pandemic. I now request Professor R.K. Mutatkar and Professor Bhushan Patwardhan to present the citation and felicitate Mande, sir. I now request Patwardhan sir to give the closing remarks.
Thank you, Professor Mande, for that brilliant presentation. Actually, we started on a very different note with our own students' memories and the gesture parents did to continue her legacy here. Thank you, Mr. and Mrs. Bakshi, for doing this. And I'm sure Subarna's legacy will continue and many more students will take inspiration and continue to work harder all over. Dr. Mande, the kind of overview which you gave in a very short time tells us that what was going on behind the screen. All of us were locked down in our own houses, but what was happening in New Delhi and how rapidly the government machinery, the private players came together and did what you just showed. That's remarkable, and your last message to go to Atmanirbharta is very, very important, because when he showed us that when they actually started, nothing was existing. Nothing was existing. So practically everything from scratch they had to do, and they did it in a record time. I'll just expand on one point because that is relevant to the school. He showed us a slide about the Ayush involvement, and so Ministry of Ayush was also involved in this, and I was chairing the National Task Force for Ministry of Ayush. And in a short period of time, with CSIR on board, we conducted clinical trials which were compliant to GLP standards. And these studies, one study only I mentioned about ashwagandha. At that time we challenged the hydroxychloroquine used, on scientific grounds. We asked these questions, and to answer that we conducted a randomized controlled double-blind trial comparing head-to-head comparison of hydroxychloroquine with ashwagandha, and we showed that ashwagandha was ahead of hydroxychloroquine in terms of prevention and prophylactic effects. During the same time, Ministry of Ayush conducted 130 clinical trials in almost 30 centers which they have. Like CSIR, Ministry of Ayush also has a council for research in Ayurvedic sciences. They have about 30 plus research centers across country. So these were multi-centric clinical trials conducted on various Ayush interventions. And you must have read that one of the effort came through Ayush Sanjeevani, that app which was created by him. Crores of people registered on that app and took advantage of prevention, not maybe in treatment, but for prevention, be it Ayush Kada or various other home remedies. People benefited from our country. So, Dr. Mande's talk tells us that the kind of capabilities we have, the scientific preparedness we have, and how our scientists can really, not only what we see in the movie Vaccine War, but what goes behind actually in science laboratories. That you showed us very, very vividly, Dr. Mande, and thank you very much for coming here and giving Dr. Deodhar oration. As was introduced by Aarti, Dr. Deodhar, maybe all of us have been trained by him, and Dr. Gupte, who is just sitting here, was recipient of Oration earlier, and he himself is one of the very prominent leading epidemiologists. And Mrs. Bakshi, you mentioned about One Health, and Suparna also carried that message outside. But Dr. Gupte is the one who started talking about One Health first. When I heard first time at that time ICMR was in a process of establishing a center for One Health and zoonotic diseases at that time. So I'm very happy that Dr. Gupta, you are also present here with us. With this, I congratulate Dr. Abhay Dugade and the entire team and faculty and students of health sciences for two reasons. One, actually inspiring young students by hosting this fellowship. And thanks Anita for mentoring this, because this was very important. As Mr. Bakshi mentioned, he had only contact with you, and the way in which you crafted the whole idea, and along with Abhay and Health Sciences faculty, is really something which many others will also follow. Many others will get inspiration for what you have done, Mr. and Mrs. Bakshi. And thank you very much. I wish you all the best. Namaskar.
The end of the program. But before we end, our students would like to present a portrait of Dr. Suparna Bakshi to her parents. So I invite the students.
Thank you, Prof. Thank you.
Please go on for taking public. Okay? You have heard, sir, just now that there are so many areas in which you can work. So make yourself capable to work in so many fields. And he said that that was not the last pandemic. So there will be more and more. If you love your country, wait for your country.
So on behalf of the Department of Health Sciences, I thank Dr. Mande, Mr. and Mrs. Bakshi, Prof sir for their gracious presence. We are also grateful to the SPPU authorities for the support. Thanks to all the guests for gathering here today. There are many eminent scientists who are here today from NCL. Dr. Mande is here, Dr. Sandeep Sarvi is here. We also have the relatives of Dr. Deodhar, Mr. and Mrs. Apte, who are here today. We thank you all, and you all are invited for tea and